Okay. Last time, first, this morning I went to the dentist. That's why my cheek is swollen like this. Okay, so I'll try not to speak too much. But okay, so we are in chapter 7. Okay, so we're studying cellular respiration. Respiration means breathing, right? But why we need to breathe to stay alive? It's to get oxygen. Okay. Then the question goes, why the cells need oxygen? Okay, that's where we are. Okay, because we can burn the sugar inside the cell. Okay, that's how we get energy. The question is how. Okay, so we're looking at cellular respiration. Okay, there's 3 stages in cellular respiration in our cells. Okay, the number 1, what is called? Glycolysis. The second one, crappy name, Krebs cycle. Third one is ETC, electron transport chain. Okay, the number 1, glycolysis happens in where? Cytosol, right? Cytosol. But the rest of it, Krebs cycle, electron transport happens where? Mitochondria. That's why we call mitochondria is the power plant of our cell. That's where they generate energy, which means ATP. Okay, so let's go through the first one. Okay. Again, summary. Some organisms rely solely on glycolysis for energy harvesting. We'll go later. For most organisms, however, glycolysis is primary process of energy extraction, only a certain situation when quick bursts of energy are required. But it is necessary first stage to Krebs cycle and ETC. In other words, you have to go through glycolysis to go to second and third stage. Okay? So again, glycolysis takes place in cell cytosol. Okay, Krebs cycle and ETC takes place in cellular organelles called mitochondria. Okay, so if you look up the page 129, okay, in your textbook, both 5th edition or 4th edition, same thing. Okay, that's the complex pathway of whole glycolysis. Okay, you got it? Page 129. Don't worry about try not to memorize all of them. Okay, you don't need to memorize. Okay, the important thing is starting. What is the starting point? Starting molecule. You start with still 마치가지 않겠어. Still start with glucose, right? Start with glucose. What's the end product? 2 molecules of pyruvic acid. Okay, and then if you can sum up, you have to add some ATP. You use ATP at the beginning, right? But at the end, you will get ATP back. All right. So the overall gain of ATP is 2 molecules, right? Anything else? Anything else than ATP is produced? You have to look up the book, though. Okay. What do you get? You get NADH from NAD plus. Okay. That's also some kind of energy that will exchange into ATP later. Okay. So that's the whole thing. Glycolysis yields 2 ATP and plus NADH. Okay, so that's the first step. But we said some organisms they don't use oxygen. Okay, in that case, you can live on this glycolysis. There should be some ways to do it. Okay, we call it what do you call it? Fermentation. It's not in your textbook. Okay, so you have to be alert. Okay, fermentation in Korean, what is it? 발효. There are two types of fermentation. One is called lactic acid or lactate fermentation. 젖산 발효. Okay. Second one is your favorite thing, alcohol fermentation. Okay. So lactic acid fermentation, where does it happen? It happens in your muscle cells. So let's say you go out and run for the 100-meter dash. You run it. Okay. So what happens is your muscle cells don't have enough time to get supply oxygen enough. Okay. So your muscle has to generate energy without having oxygen in the cell. Okay. So what happens is from glycolysis you have pyruvic acid, right? What happens is pyruvic acid is changing to next molecule called lactic acid. That's why it's called lactic acid fermentation. Okay, you don't need any oxygen. But what happens is if you change from lactic acid to pyruvic acid to lactic acid, what happens is you got this NADH from glycolysis, right? But you have to use this NADH and then back to NAD+. So in this case, NAD+ is shuttling. It's just like taxi. It's changing electrons. Okay? So you start with glucose to end up with lactic acid. But doing so, you can generate 2 ATP. That's all. But you can do it without oxygen. That's how your muscle cells do when you do running, fast running. Okay? So that's why next day you get sore muscle, which means lactic acid is in your muscles. So you have to get rid of it at some point slowly. Okay? Second type of fermentation is called alcohol fermentation. Okay? So how does it happen? Very similar. From pyruvic acid, which is the product of glycolysis, right? Pyruvic acid going to change into acetaldehyde. Okay, this one is a little bit, it's a C2 molecule. Have you heard about acetaldehyde causing you a hangover? No? 선전에 나오는데 그 다음날 술 먹고 그 다음날 아세탈데하이드를 잡아라 뭐 이런 얘기 나오는데, right? So this is the molecule, okay, that will give you hangover after heavy drinking. Okay, this is why. Okay, so pyruvic acid becomes acetaldehyde. Okay, between become acetaldehyde become ethanol. Okay, that's when you can recycle NADH into NAD, so you can shuttle it. Okay, so the final product is ethanol. So this is ethanol fermentation. This is why all the wines cannot go over higher than 13 or 14% of alcohol. No? So the highest alcohol contents of wine, right, is only 13% or 14%. Why? The living yeast Saccharomyces cerevisiae, yeast is doing all this fermentation. Okay, if the alcohol concentration goes higher, it will kill yeast. So they don't make ethanol anymore. Okay, that's why the wine is highest alcohol contents is 13% or 14%. But if you want to make higher liquor, you can boil it and capture the wine, and then you can have 30%, 40% alcohol. That's the cognac. It has the flavor of wine, but it's much, much stronger alcohol. Okay? So if you drink alcohol, if you drink alcohol, what happened? It has to go through your metabolism. So it's going backwards, acetaldehyde and pyruvic acid, and then they can go through Krebs cycle or TCA cycle to burn it. Okay? But if you drink too much, what happened? It's stuck here, acetaldehyde. That will give you next day morning headache, hangover. Okay? And also, some people, like Asians, they don't have enough enzyme to convert ethanol to acetaldehyde. How do you know? If you drink a little bit, your face turns red, that means you don't have the enzyme, which means you shouldn't drink. Okay, that's the warning sign. If you keep drinking, you'll pass out. Okay, so that's the side story of fermentation. But that's the side story. The main stage, first stage of this cellular respiration is what? Glycolysis. Okay. All right. So this is overview. Okay. Even if you forget about everything, you should remember this, right? So the first one is glycolysis. From glucose, you go 2 molecules of pyruvic acid. In doing so, you get 2 ATP. But at the same time, you get some 2 NADH because it has electrons, so it has potential energy. Okay. The second step is Krebs cycle, and third step is ETC. But you cannot go directly from pyruvic acid to Krebs cycle. There is a conversion a little bit. Okay, we'll come back to point. So Krebs cycle still gets little ATP, 2 ATP, but you get lots of electron, 6 NADH and 2 FADH2. It's the same thing. It's an electron carrier. It's just different color of taxi that carries electron. Okay? And at the end, all this NADH and FADH is riding a roller coaster, okay? Dropping electron energy, okay? And that will generate ATP. How? We'll come back, okay? So most ATP comes from the third stage, which is called ETC, electron transport chain. Questions at this point? Okay, let's go. All right, so we did glycolysis. Okay, so again, you start with glucose, end up with 2 pyruvic acid, and then you get 2 ATP, although it says 4, but you used 2 molecules. That's why I end up with 2 ATP and 2 NADH. Okay, the second step, Krebs cycle. Okay, so I told you glycolysis starts from, it's happening in cytosol, right? But then pyruvic acid has to move to inner compartment of mitochondria, okay? You need conversion at this point, okay? So what do you have to do? From pyruvic acid to called acetyl-CoA. It's a little bit strange name, but you know. Acetyl-CoA is the molecule that enters Krebs cycle, okay? So Krebs cycle generates lots of electrons, okay? You don't have to remember 6 or 2 or whatever, okay? 2 molecules of ATP, okay? It's very complex, okay? If you can memorize, that's good. You don't have to, okay? Unless you are a bio major, thing. Okay, so you start with acetyl-CoA, okay? You run this cycle. So what happens is some places they generate NADH or FDH, okay? And some place you will generate ATP, okay? You run cycle, okay? Krebs cycle. Right, the last stage, and the question is, how you generate ATP, okay, fine, but you generate this FADH or NADH, and how can you generate ATP from that? Okay, that's the question, and that's third stage. And A lot of explain, you can read it. So this electron transport system, it's like stepping down stairs. Okay, why? You don't just jump from one, okay? By doing step by step, stepping down, you can use little amount of energy at a time, okay? But each step, using that energy, what do? You kick out hydrogen ion, H plus ion, to one side, okay? Do you remember the gradient difference or concentration gradient, right? So what happened is you pumping this H plus to one side of membrane and generating gradient concentration gradient. In other words, you pile up this hydrogen ion, okay? You pile up so high this hydrogen ion, they want to come back, okay? When they want to come back, that will generate ATP. Okay? In other words, each step of electron transport system and kicking out, okay, hydrogen ion, and you pile up hydrogen ion. When they want to come back, that will make ATP. It looks like this. Okay? So each step of this NADH electron transport system, a lot of membrane protein sits on this membrane, okay? They trap those little energy steps, okay? So what they do, they pump this H plus into one side. This is mitochondria inner compartment, okay? So this is mitochondria outer compartment. You remember, mitochondria had two double membrane, yeah? Okay. So you kick out this hydrogen ion, okay? Each step, so NADH or FADH, and the final acceptor of this electron is important. Who's accepting electron? Yes, that's the oxygen. That's why we have to breathe, okay? That's why we use oxygen, okay? So the final electron will trap the electron and become water, okay? So you pumped out all the proton, H plus ion. And how do you make ATP out of it? So later, okay, this part has much higher concentration of H plus, okay, and they want to come back, okay. They can only come through this big enzyme called ATP synthase. ASE ends with ASE, so it's an enzyme, right? Big protein, and when the protein comes back, what do you have to do? From ADP to ATP. They can generate ATP. Okay? So this the whole story. You start with glucose. This is what you eat. Okay? And it will break down in cytosol. The first step is glycolysis. It will break down into pyruvic acid. And pyruvic acid enters mitochondria. Okay? It changes into acetyl-CoA. Acetyl-CoA goes around Krebs cycle. They'll generate ATP, but they'll generate lots of NADH, FADH. Okay? Those electron molecules go through stepping down. They use energy, pumping out proton. Okay? The final acceptor of this electron is oxygen. That's why we need oxygen. Okay? And then they pumped out proton, generate concentration gradient. Okay? When they comes back, they'll generate ATP. Okay? You got the big picture? Okay? Also, you got sleepy, right? All right, that's it. So what happened to if you eat other than glucose, if you eat meat, okay, they have protein. Okay, what happened? So three major nutrients. Okay, carbohydrate, we know the sugar, that's glucose, right? So if you eat proteins or fat, they can break it down to different molecules, and they can enter glycolysis, or some of them go directly to Krebs cycle. Okay, so they have all the ways to change it, to go through it. So the final thing is the whole thing. Okay, you have to use oxygen. That's why it's called cellular respiration. The final step, the electron transport chain, is the jackpot. They'll generate lots of ATP. Okay? All right. Okay. Some more nonsense quiz then. Okay. A guy walks into a bar. Okay. And he asks a glass of water. And bartender pulls out a gun. The guy said, "Thank you," and he left. What's going on? Huh? What? I'll repeat one more time. Okay, you have to listen carefully. A person walks into a bar. Okay. And he asks a glass of water. And bartender pulls out a gun. And the guy says, "Thank you." And then he left. What's going on? No? No idea? Veronica? Nope? Yes? Great. So he was hiccuping. So he asked a glass of water. The guy pulls out a gun, scared him out of it, and he said, "Thank you," and then he left. All right? One more. This guy is coming home, and he knows there are two guys with masks on are waiting for him. What's going on? This guy is coming home, and he knows there are two guys waiting for him with masks on. One more time. He's coming home. He knows there are two guys, two persons, waiting for him with masks on. What's going on? Photosynthesis? It's a baseball. This guy is running around third base. He's running for the home. There's an umpire and a catcher is waiting for him. Okay? All right, good. All right, let's go to photosynthesis. Okay, do we have a movie? No, we don't have a movie. Okay, so we start. Cellular respiration is you're breaking down glucose. Okay, now the question is, how do you make glucose? Okay, where does glucose come from? Okay, so all the nutrients that we eat originally coming from the sun's energy. Okay, if you don't have plants or if there is no photosynthesis, we have nothing to eat. Somebody said, we can eat meat. Yes. What the animals eat? They eat grasses, right? And where does grass come from? Coming from sunlight. Okay. Photosynthesis has made possible life as we know it on Earth because the organic material produced in photosynthesis is the source of food, our food. Okay? So what is the photosynthesis? You need light. Okay, that's why it's photosynthesis means you're making something. All right? So what are you making? Sugar. From where? Okay, from carbon dioxide. And also photosynthesis generate the oxygen. That's great, right? We have to breathe oxygen. And plants will generate oxygen as a byproduct of photosynthesis. Okay, so this is great. Okay, So sunflowers or giant kelp. Remember the movie you saw in Harry Potter? Yeah, you see the underwater plants. Okay, and then some bacteria, cyanobacteria, can do the photosynthesis. Okay, they have thing common. Okay, they all have organelle, cellular organelle called chloroplast. Okay, chloroplast. Don't confuse with chlorophyll. Chlorophyll is a pigment, small pigment. Okay, you can find in chloroplast. Okay, chloroplast is organelle. Okay, cellular organelle. 엽록체, chlorophyll, 엽록소. Okay, different. All right. So why most of the leaf is green? Okay, because they absorb, right? Absorb red and blue, and they bounce off this green color. That's why we see as a green. Okay, so in other words, to do the photosynthesis, best way is to absorb blue and red. Okay, that's why most of plants their leaves are green. Okay. Okay, so this is how we see it. Okay, so photosynthesis is you using water and you using carbon dioxide, which you breathe out. Okay, plant will use these two, and they will synthesize sugar. Then they'll give out oxygen. It's great, right? So we eat this sugar, and we burn with oxygen, and we can use ATP. And now question comes. Which comes first? Is it photosynthesis or cellular respiration? Photosynthesis or cellular respiration? Right. So we believe that in old days, very, very old days, the Earth atmosphere didn't have oxygen until this photosynthesis comes up. Okay? So they did photosynthesis and they get rid of oxygen. Okay? Now animal evolved, or small animal or small bacteria evolved to use oxygen to generate more and more ATP. Okay? So the photosynthesis comes first. Okay? And then cellular respiration. Okay? So basically, so this chapter will deal with how can you use this carbon dioxide and water, and the energy comes from sunlight. How can you make sugar? Okay, that's the whole story of photosynthesis. Where this thing happen? Okay, it happens in plant, it happens in leaves. So we have to know some structures of leaves. Okay, the word is called stomata. Okay, stomata is a little opening. The back side of the leaf, they do gas exchange. Okay, that will get rid of oxygen, and they will uptake carbon dioxide. Okay, that's why if you walk in the woods, you feel very refreshed, right? Why? Because there's lots of oxygen around. You breathe, you feel nice. Okay, so that is structure called stomata. Okay, this is the leaf. There are some cells, mesophyll cells, whatever, and mostly mesophyll cells are doing photosynthesis. Okay, if you look at this is plant cell. Okay, within plant cell, this is called chloroplast. Again, like mitochondria, chloroplast has two membrane, double membrane, outer membrane, inner membrane. Okay, inside chloroplast you'll see some choco pies, right? Choco pies stacked up. Okay, it's called thylakoid membrane. Okay, and inside thylakoid membrane and outside. Outside is called stroma. Don't get confused with stomata. Okay, stroma is the inner space of chloroplast. Stomata is the opening. Okay, opening for gas exchange. Got it? Okay, still sleepy? Okay. Yeah, I need a break. So photosynthesis can be divided into two stages. Remember, cellular respiration was divided into three, right? It's a little simpler. Photosynthesis, there is a light and there is no light. Okay, so the first stage is called light reaction. The second stage used to call it dark reaction. Instead, these days they call it Kelvin, not Klein, Kelvin cycle. Okay, that's the person who identified this cycle. Probably he got Nobel Prize at some point. Okay, so before we go to next chapter, this is about photosynthesis. Okay, have you seen a movie called Trends? 우리나라에 안 들어왔나? No. Okay. How about Les Miserables? Yeah, did you see it? No? 안 본 사람은 어디서 비디오 구해서 한번 봐야 될 것 같아. 내가 지난번 출장 가면서 비행기에서 또 봤는데 너무너무 좋아. So you should see. Okay? So the books that I want to recommend, it's called Life Lessons. In Korean, 인생 수업. Okay? It was written by Elizabeth Roth Quibler and one more person, but yeah. And then translated by Yu Shi-hwa. Okay? It's a good book. So this doctor interviews all the terminally ill patients. Okay? They're facing death. Okay? They know they're gonna die. Okay? So the question is asking them, what is life? Okay? Because we think we can live forever. Okay? You don't want to think about death, right? But this patient has to think about death. And this will teach you what the meaning of life is. Okay, it's a good book, so you should read either in English or in Korean. Okay? All right, so going back to photosynthesis. Okay, so you can divide this photosynthesis into two stages. Number one, photo. Okay, there is a light. So light reaction. Synthesis, you're making something. That's Kelvin cycle. Okay, so the first reaction is light reaction. What's going on with first light reaction? There is a light, sunlight comes. What it does is sunlight will split the water molecule. Okay? If the sunlight splits water molecule, what is water molecule? H2O, right? Splits it, and then oxygen comes, byproduct, and what happens is it will elevate the electrons. Okay, it will pump up electron. Do you remember that electron that we captured by burning glucose? What happened? You captured the electron. What do? You're going down, down, down. Right? That's exactly the same thing. Using the light from, sunlight, you split the water molecule, the oxygen will come, and then electron will pop up. Okay? And you're just coming down the electron, you capture those energy. What do? You transfer those electrons, right? Eventually, what do? You pump out protons somewhere. Pump in or pump out, whatever. Okay? And then you generate the gradient, concentration gradient, and it will generate what? ATP. That's the first light reaction. Okay? So the first stage called light reaction, electrons derived from water, okay, by the power of sunlight. It will split this water molecule. Okay? So electron carrier has a little bit different name from cellular respiration. What was the name of electron carrier? The cap, taxi, NAD plus or NADH, right? There's one more letter in this case. It's called NADPH. Okay, it's basically same. They'll carry electron. They'll carry electron down, 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 and then use that energy to pump out something, pump out proton. Okay. The second part is synthesis. Okay, you have to make something. What is the starting molecule? It's called carbon dioxide, CO2. And eventually you will make sugar out of it. What do you need? From small molecule, you go to big molecule, what do you need? You need energy, right? Where this energy comes from? ATP. That's you made in light reaction. Okay? It's very simple if you look at the big picture. Okay? But if you go in detail, it's a little bit more complicated, but you don't want to know all about them. Okay? So again, the antenna molecule is called chlorophyll. Chlorophyll. Antenna is just capturing the light energy. Okay? That's all you have to know. You don't have to know anything else. It's just like sunlight comes in, then chlorophyll, the pigments will capture this light, and this light energy will split water molecule, oxygen will come out, electron activated. And you trap this electron by NADPH, and then you come down, and then you make ATP out of it. Okay, that's basically it. Okay. So two actions of great consequence take place in the light reaction. What is it? Water is split, and electrons and oxygens are produced. The oxygen is very important because we have to breathe oxygen, right? And then electron, it activated, and we can trap those energy to produce ATP, right? And that's here. Where this thing happen? Chloroplast is cellular organelle in plant cells. Okay. Now we're looking at, this is chloroplast. Okay. It has outer membrane, inner membrane. All the action happens within this inner membrane. Okay. And this look like choco pie. Their membrane is called thylakoid membrane. Okay. It's called thylakoid. Okay. In this membrane, all the proteins are sits in the membrane. Okay. So what happens is the sunlight comes in, it will split the water molecule. Okay. It will split the water molecule. You get oxygen. Okay. And then what happened? So the electron activated. Okay. Again, this is very similar like electron transport chain, like what we saw in mitochondria. Okay, same thing. You step down these electrons. And what will happen is, by doing so, the textbook doesn't show, but it has to pump proton to somewhere. Okay? And those proton concentration gradient, and they want to go out again. And then what will happen is missing here is the enzyme called what? ATP synthase. Okay? That sits in there, it'll generate ATP. Okay? So at the end of this light reaction, what do you got? You got plenty of NADPH, which is carrying electron. Okay? And it didn't show here, but lots of ATP. So now you're ready for Calvin cycle. In other words, you can synthesize sugar. Okay? So this electron transport system will, at the end, will generate NADPH, and also will generate lots of ATP. Okay. The second one is more complicated, all right? It's called Kelvin cycle. The person who identified all these steps, okay, is Kelvin, okay? Kelvin cycle is the second stage of photosynthesis, okay? What is it? It's a synthesis step. In other words, you have to make something from carbon dioxide. What are you making? Sugar. But you don't make glucose right away, okay? There's a whole step of it, okay? What is the result of this Kelvin synthesis? It's called G3P, something like has 3 carbon and then has phosphate in it. We'll go through it, okay? Again, it's very complex pathway. You don't have to memorize all of them. But the overall thing is you add ATP and NADPH and you will make from carbon dioxide to sugar. Okay? So you have to remember a few things. Okay? The first molecule this CO2 will meet is 1, 2, 3, 4, 5 carbon molecule. Okay? It's called RUBP. Okay? 5 carbon molecule will meet one more carbon, which is carbon dioxide. Okay? And then it will become carbon 6, right? It should, if it is glucose, it would be great, right? Just one step. No, that's not the way. Okay? So you split this 6 carbon into 3 and go around. At the end, the coming out G3P, in other words, carbon 3 molecule. At some points, you have to combine these two to make glucose. So important thing is the first molecule you will meet, this CO2 will meet, is called RUBP. Okay? The enzyme that combine this reaction is called Rubisco. Okay? Rubisco is the most common, most abundant enzyme in the world because photosynthesis is most basic things. Every plant has it, and this enzyme is most common enzyme. But it has some defects in it. We'll come back. Okay? What does this enzyme do? It will combine this carbon 5 molecule, RUBP, called RUBP, and then it will put one more carbon molecule, CO2, into it. I'll make 6. Okay? And then what is the outcome of this Kelvin cycle? It goes around, and it will come G3P is the product. Okay? Yep.
At the end of World War II, it was known that plants made their own food, high-energy carbohydrates from carbon dioxide, but the process remained unclear. Led by University of California, Berkeley chemist Melvin Calvin, researchers unraveled the mystery of photosynthesis with the use of radioactive isotopes, namely carbon-14. With carbon-14, one can tag carbon dioxide to track its course during photosynthesis. Calvin and his colleagues chose algae for their photosynthesis experiment. Like all plants, algae use carbon dioxide to produce food. The trick of the experiment was to allow the algae to perform some photosynthesis using carbon-14, then stopping the process at different points to observe what the carbon-14 becomes. In order to find the carbon-14, two-dimensional paper chromatography was used. First, the material of interest, in this case a sample of algae, was placed on the paper in solution form. The solvent then moved through the paper, separating out the components of the original material. The components are shown as green dots, but in the experiment, the components were not visible. Next, paper chromatography in the second dimension is done. The paper is rotated 90 degrees and put in a second solvent. The component compounds are separated in the second dimension. Some of the compounds contain the radioactively labeled carbon. This process leaves the components drawn out in two directions by both solvents. Setting X-ray film tightly on top of the chromatography paper for a long period of time, usually about 2 weeks, causes the radiation from the carbon-14 to expose the film. Spots show up on the film, showing the location and concentration of carbon-14. It took Kelvin and his colleagues 10 years to properly interpret these spots. By this process, what we now call the Kelvin cycle, conversion of carbon dioxide to food, was finally understood.
Did you get the idea? No? No. Okay, that's fine. So Kelvin and his colleagues spent 10 years to just go figure out the cycle. And you don't want to even memorize it, right? So how do you do it? So first, plants uptake CO2, right? And then next question is, where does CO2 goes? Which molecule will take up this CO2? So the thing is, you label this carbon with radioisotope, okay, that has energy. It's like when you're having a chest X-ray, okay? So how do you get this X-ray film developed? You go like this, and then X-ray go through, and then it will develop the film, right? Same thing. This little carbon dioxide has radioisotope. So it has energy will shoot out, and then that will develop the film, okay? So the idea is where this carbon molecule in carbon dioxide goes next. Probably he will found the carbon-6 molecule. And then if time goes, that will split into carbon-3 and going on. And he just followed. It took 10 years. Okay? So it takes hard work of scientists to just show this pathway. It took 10 years to draw that pathway, right? And we don't even want to memorize it. All right. So G3P is the outcome product of this synthesis, Kelvin cycle. Okay, that's all you have to remember. Okay. Now, photorespiration and C4 pathway. This is a little bit thing new. Okay. We know photosynthesis, right? But we know cellular respiration. The question is, what is this photorespiration? Okay, so the trick is, okay. So I told you this enzyme is most common, okay, Rubisco, okay. Its job is take this carbon dioxide and put it up here and make 6 carbon molecule, okay, this Rubisco. But it has a little bit problem. It has a little bit sloppy job. It doesn't have good, job. So what happened is, again, plant cell also have to do cellular respiration. In other words, they have to also make energy too. Not only synthesize sugar, but at some points they need ATP for something else, right? What happened is this enzyme is not very specific for carbon dioxide. Okay? Sometimes if there is oxygen around, it will use oxygen. Then what happened? It's not building up something, it will break down something, right? That's the respiration. In other words, if there is oxygen around here, okay, and then it will break down. That's why it's called photorespiration. Okay, so it's a bad thing to happen for plant cells. Okay, so why this thing happen? Because its plant is not like animal breathing in, breathing out, breathing in, breathing out. You take CO2. You take oxygen. You breathe out carbon dioxide. Plant cannot do it. How can you do it? You just open up the air hole, which is what it's called, stomata. Just open up, and you exchange with oxygen and carbon dioxide. Okay? So that's the problem. So in other words, at a certain stage, oxygen and carbon dioxide mixed up. Okay? The enzyme cannot always find carbon dioxide and put it in. That's the problem. That is called photorespiration. Okay? It's especially a problem for plants during the hot summer day. If they open up the stomata to get carbon dioxide, okay, then oxygen can also come in and binds to that molecule because rubisco is such a sloppy enzyme. Okay, then it's a problem. So how this plant evolved to solve that problem? It's called C4 pathway. Okay, in plants, the enzyme rubisco frequently binds with oxygen rather than the carbon dioxide. Okay, a process called photorespiration. Okay, so how do you prevent this one? It's called C4 pathway. Where this C4 comes from? What do you mean by C4? There's, right, carbon molecule with 4 carbon. Okay, so remember Kelvin cycle? If it is regular Kelvin cycle, what happened? CO2 will meet 5 carbon molecule, right? But what do you do in C4 pathway? Okay, so they'll use 2 rooms. They'll use separate rooms, okay? So in one room, okay, they collect more CO2, okay? How do you collect? You have molecule that has higher affinity for carbon dioxide, okay? Use that molecule, trap carbon dioxide, and move that carbon dioxide into the next room, and then just give it to it, okay? So the problem is capturing carbon dioxide specifically, right? So this case, the C4 molecule, okay? So C3 probably. C3 molecule captures CO2 and then it will become C4 molecule. Okay? And then C4 molecule gives out carbon dioxide to next room. And then they can run the Kelvin cycle. Okay? So the plants like corn, okay, they can use this C4 pathway. So they can do more photosynthesis during the hot summer day. They can open up stomata, they can capture carbon dioxide more specifically and use it. Okay, it's called the C4 pathway. What about plants live in desert? Okay, desert, such as cactus, 선인장. Okay, as soon as they open their stomata during the day, they will be dried out. Okay? So what do? We have time difference. In other words, during the night, they open up stomata, they can collect the CO2, and then next day there's a sun, they'll do synthesis. Okay? It's called CAM photosynthesis. What CAM stands for? You don't want to know. Caselucin acid metabolism. Don't worry about it, okay? It's just called CAM, CAM pathway, or CAM photosynthesis, okay? So this one, using time difference. Okay, during the night, they open, they capture carbon dioxide. Next day, they do the photosynthesis, synthesis Kelvin cycle, okay? It's called CAM photosynthesis. Which plants use this one? Cactus, sugar cane or whatever, okay? Such as cacti employ another form of CAM. Okay, so we talked about photosynthesis, right? Photosynthesis is divided into 2 groups, 2 stages. The first one is what? Photo, light reaction. You use the light, split the water molecule, oxygen will come out, electron pops out, you use electron energy and trap, and then you produce NADPH. At the same time, you produce lots of ATP. Okay. Second stage is called synthesis or Kelvin cycle. Okay, so you start with carbon dioxide. The first molecule it will meet is called, what is it? RUBP. Okay, the enzyme that combines this one is called Rubisco. Okay, final product is 3GP. That's the kind of sugar. Okay, that's the regular Kelvin cycle, but it has some problem, right? Rubisco can not only capture carbon dioxide, it can sometimes capture oxygen, which we call it photorespiration. Okay, how this plant solve this problem? It's called C4 pathway or CAM photosynthesis. C4 pathway is using another compartment. They use one more room to capture carbon dioxide. CAM plants, they use time difference. They open up stomata during the night, they capture carbon dioxide. Next day, during the day, they do the synthesis. Okay? So I guess we'll stop here. Okay? Next week, we have to go, what? Chapter 9 and chapter 10. And week after next week, what's waiting for us? Midterm exam is waiting for us. Okay? It's all multiple choice probably. And about 10 questions per chapter. That is total what? About 100. Okay? So each solve the problem in summary. So next week I'll go through not only chapter 9 and chapter 10, I'll go from the beginning to the end, the review. Okay? So I'll see you next week. So I have some homework printed out back. You can, if you didn't print out chapter, what is it? 8, 9, you can have it. There's also summary in the same file.